The overall goal of this protocol is to enhance the incorporation rate of non-canonical amino acids in mammalian cells to tackle biological questions in live cell settings, including mapping of protein-protein interaction surfaces and bioorthogonal labeling of surface receptors.
Using amber codon suppression, different non-canonical amino acids are incorporated with different efficiencies. We established a fluorescence assay to easily compare non-canonical amino acid incorporation rates in mammalian cells within a short time.
The systematic incorporation of a photo-crosslinking non-canonical amino acid throughout GPCR surfaces allows mapping of ligand binding sites and provides precious information of the intact receptor directly from the live cell.
By incorporating last-generation non-canonical amino acids for bioorthogonal chemistry, we achieved site-specific, super-fast, catalyst-free labeling of GPCRs with small blind fluorophores for high-end imaging applications in live mammalian cells.
First, seed 600,000 HEC293 cells in 2 milliliters of complete growth medium per well in 2 6-well plates. Prepare as many wells as the number of samples and 2 additional wells for the wild-type EGFP and a mock transfected sample, respectively. 1 hour prior to transfection, add the appropriate amount of freshly prepared non-canonical amino acid stock solution to all wells for a final amino acid concentration of 0.25 to 0.5 millimolar. To transfect the cells, mix 1 microgram of plasmid DNA encoding for the synthetase tRNA pair to be tested with 1 microgram of reporter plasmid DNA in a